can be thought as a bag in which the chemistry of life occurs. The activity going inside the cell is therefore separated from the environment outside the cell. The bag or cell is surrounded by a thin membrane. The membrane is an essential feature of all cells because it controls exchange between the cell and its environment. It can act as a barrier, but it can also control movement of materials across the membrane in both directions. The membrane is therefore described as partially permeable. If it were freely permeable, life could not exist because the chemicals of the cell would simply mix with the surrounding chemicals by diffusion, and the inside of the cell would be the same as the outside. There are two types of cells. During the 20th century, scientists studying the cells of bacteria and of more complex organisms such as plants and animals began to realize that there were two fundamentally different kinds of cells. Some cells were very simple, but some were much larger and more complex. The complex cells contained a nucleus, plural nuclei, surrounded by two membranes. The genetic material DNA was in the nucleus. In the simple cells, the DNA was not surrounded by membranes but apparently free in the cytoplasms. Organisms made of cells with membrane-bound nuclei are now known as eukaryotes, while the simpler cells lacking membrane-bound nuclei are known as prokaryotes. Eukaryotes are thought to have evolved from prokaryotes more than 2 billion years ago. Prokaryotes include bacteria, eukaryotes include animals, plants, fungi, and some other organisms. The study of cells has given rise to an important branch of biology known as cell biology. Cell biologists study cells using many different methods, including the use of various types of microscope. There are two fundamentally different types of microscope, the light microscope and the electron microscope. Both use a form of radiation in order to see the specimen being examined. The light microscope uses light as a source of radiation, while the electron microscope uses electrons for reasons which are discussed later. Features that animal and plant cells have in common. Cell surface membrane. All cells, including those of both eukaryotes and prokaryotes, are surrounded by a very thin cell surface membrane. This is also sometimes referred to as the plasma membrane. As mentioned before, it is partially permeable and controls the exchange of materials between the cell and its environment. Nucleus. All eukaryotic cells contain a nucleus. The nucleus is relatively a large structure. It stains easily and is therefore very easy to see when looking down a microscope. The deeply staining material in the nucleus is called chromatin. Chromatin is a mass of coiled threads. The threads are seen to collect together to form chromosomes during nuclear division. Chromatin contains DNA, the molecule which contains the instructions that control the activities of the cell. Inside the nucleus, an even more deeply staining area is visible, the nucleolus. This is made of loops of DNA from several chromosomes. The number of nucleoli is variable, 1 to 5 being common in mammals. One of the main functions of nuclei is to make ribosomes. Cytoplasm. All the living material inside the cell is called the protoplasm. It is also useful to have a term for all the living material outside the nucleus. It is called cytoplasm. Therefore, cytoplasm plus nucleus protoplasm. Cytoplasm is an aqueous material varying from a fluid to a gel-like consistency using a light microscope. Many small structures can be seen within it. These have been linked to small organs and therefore known as organelles. An organelle can be defined as a functionally and structurally distinct part of a cell. Organelles are often, but not always, surrounded by one or two membranes so that their activities can be separated from the surrounding cytoplasm. Organizing cell activities in separate compartments is essential for a structure as complex as an animal or plant to work efficiently. Mitochondria. The most numerous organelles seen with the light microscope are usually mitochondria. Mitochondria are only just visible using a light microscope. Videos of living cells taken with the aid of a light microscope have shown that mitochondria can move about, change shape, and divide. They are specialized to carry out aerobic respiration. Golgi apparatus. Use of Golgi stains containing silver resulted in the Golgi apparatus being discovered in 1898 by Camillo Golgi. The Golgi apparatus collects and processes molecules within the cells, particularly proteins. Differences between animal and plant cells. One of the structures commonly found in animal cells which is absent from plant cells is the centriole. Plant cells also differ from animal cells in possessing cell walls, large permanent vacuoles, and chloroplasts, cell walls, and plasmodesmata. With a light microscope, individual plant cells are more easily seen than animal cells. This is because they are usually larger and, unlike animal cells, are surrounded by a cell wall. Note that the cell wall is an extra structure which is outside the cell surface membrane. The wall is relatively rigid because it contains fibers of cellulose, a polysaccharide, which strengthens the wall. The cell wall gives the cell a definite shape. It prevents the cell from bursting when water enters by osmosis, allowing large pressure to develop inside of the cell. Cell walls may be reinforced with photoextracellulose or with a hard material called lignin for extra strength. Cell walls are freely permeable, allowing free movement of molecules and ions through to the cell surface membrane. Vacuoles. Vacuoles are sac-like structures which are surrounded by a single membrane. Although animal cells may possess small vacuoles such as phagocytic vacuoles, which are temporary structure, mature plant cells often possess a large permanent central vacuole. The plant vacuole is surrounded by a membrane, the tonoplast, which controls exchange between the vacuole and the cytoplasm. The fluid in the vacuole is a solution of pigments, enzymes, sugars, and other organic compounds. Chloroplasts. Chloroplasts are organelles specialized for the process of photosynthesis. They are found in the green parts of the plant, mainly in leaves. They are relatively large organelles and so easily seen with a light microscope. It is even possible to see tiny grains inside the chloroplast using a light microscope. These are the parts of the chloroplast that contain chlorophyll, the green pigment which absorbs light during the process of photosynthesis.
Is it thought that life arose on Earth around 4 billion years ago? The endosymbiotic theory states that some of the organelles in today's eukaryotic cells were once prokaryotic microbes. In this theory, the first eukaryotic cell was probably an amoeba-like cell that got nutrients by phagocytosis and contained a nucleus that formed when a piece of the cytoplasmic membrane pinched off around the chromosomes. Some of these amoeba-like organisms ingest prokaryotic cells that then survive within the organism and develop a symbiotic relationship. Mitochondria form when bacteria capable of aerobic respiration were ingested. Chloroplasts form when photosynthetic bacteria were ingested. They eventually lost their cell wall and much of their DNA because they were not of benefit within the host cell. Mitochondria and chloroplasts cannot grow outside their host cell. Evidence for this is based on the following. 1. The first, chloroplasts are the same size as prokaryotic cells, divide by binary fission, and like bacteria have FTS proteins and at their division plane. The mitochondria are the same size as prokaryotic cells, divide by binary fission, and the mitochondria of some protists have FTS homoglomes at their division plane. Second, mitochondria and chloroplasts have their own DNA that is circular, not linear. The third, mitochondria and chloroplasts have their own ribosome that have 30S and 50S subunits, not 40S and 60S. The fourth, several more primitive eukaryotic microbes, such as Giardia and Trichomonas, have a nuclear membrane but no mitochondria. Although evidence is less convincing, it is also possible that flagella and cilia may have come from spirochetes.